Hey guys, Adam here again at Society for Neuroscience Conference 2012 in New Orleans, and I'm going to be talking to you guys today about schemas. So, schemas are kind of constructs in memory that allow you to kind of flexibly apply some of your semantic information. And so, semantic knowledge, just to give another definition before we really get going here, semantic knowledge is memory for facts, memory for things that have been kind of dissociated from their original source. So, in a simple example of semantic memory, you can remember the fact that in 1776 the Declaration of Independence was signed. And you know that piece of information, but you probably can't remember exactly when you learned that piece of information. And so that's an example of a semantic memory. It's a memory for a piece of information where the information has kind of been dissociated from the actual source or context of the original encoding. But semantic memory is not a schema, not on its own anyway. A schema is an interrelation of multiple pieces of semantic information. And so a schema is really stored as a set of associations and doesn't necessarily exist in any individual piece of semantic information. And an example of a schema would be a simple script, for example, for what to do when you go out to eat. So you know when you walk into a restaurant that you are supposed to wait for the hostess or host to seat you, and then you know that when you get there you can just wait until the waiter gets to you and talks to you, and you understand kind of the general way in which that interaction is supposed to work, and you know how that's supposed to work because you have all of these semantic representations for all of the pieces of information necessary to make all of those judgments in the moment, and all of them are interrelated into this schema of what it's like to go out to eat, basically. And so a schema allows you to function more smoothly in the world, and a schema is really an important way of kind of organizing information in mind. And so in a session earlier today about the reemergence of schemas, there was a particularly interesting talk that I'm going to talk to all of you about by Richard Morris. The title of this talk was Schemas and Animals, Bridging the Gap. And so he was trying to do a study of schemas in rats. And so you can imagine this is going to be very difficult. So the goal here is to try and show that these rats have the ability to learn these kind of these associative networks of semantic information. And so we're going to take a look at how he did that. So I have up on screen now a diagram of his basic experimental apparatus. And so you can see this is just a large open field, about 4 feet by 3 feet. And I'm just guesstimating on that based upon a couple pictures that he showed us during the talk. And so you can see around the box there are 4 start boxes. And so the rat would always start in one of these 4 start boxes. And then there are 8 little wells indicated by these circles. And I've labeled 2 of them as A and B. And in each of these wells there would just be some sand. And the sand could have a food reward buried within it. And so each one of the wells was associated with a specific food reward. So for example, well A right here in the middle, could be associated with with strawberries, for example. So well A will always either have nothing or it'll have strawberries. And so when the rat is in the start box, the rat will be exposed to a scent. Say the rat is exposed to the scent of strawberries. The rat then knows that only well A will have a food reward, and so the rat should go immediately to well A. Possibly the rat would be exposed to the smell of bananas, and let's say bananas are associated with well B. That would mean then that the rat would then know that only well B is going to have food, and it would proceed like this for all eight of the wells. Always only one well, and this is the key part. Always only one well would have food, and so the indication of learning is how quickly does the rat go from the start box immediately to the well that is the rewarded well, the one well that has food, and the rat should know this just based upon the smell to location association. And so over time, the rats are able to learn this task. And here I'm putting up a chart of the rats' performance over trial sessions. And so over time, you can see that the rats are able to learn this task, and they get well above chance. The thing is, though, that this takes about 6 weeks. So to do about 20 trial sessions takes about 6 weeks. These are long, tedious trial sessions, and it takes a long time for the rats to learn these associations. So this is not a novel, or this is not an easy thing for the rats to learn. It takes a long time. However, if the rats have learned a schema to represent the overall map of the open field in which the wells are placed, then it should be possible for them to just simply update this schema with new information, and that should be much easier than actually learning the whole schema to begin with. And so to test this, they asked of the rats, they asked, can the rats learn new locations within this already existent schema? And so to do this, they simply changed the location of two of the locations. They simply changed two of the wells, so A becomes A prime here and B becomes B prime. And in addition to that, they're also actually changing what smell is associated with now A prime and what smell is associated with B prime. So maybe A prime, we remember A had strawberries before, and maybe now A prime is going to have pistachios. And B had bananas before, and maybe now B prime is going to have apples, something like that. And so the thing is, if the rats have no schema for the overall map, then it should take them a full 6 weeks again to learn this. But what Richard Morris found, was that it actually only takes a single trial. The rats are able to learn these new associations extremely quickly. And so that suggests that there must be some sort of a schema representation in mind of the overall map for this for these food and well associations. They've actually got this map of the overall field in mind, and they're able to just simply do a quick update of that map in order to do this new learning. So that suggests that schemas do exist for these rats. So another question now is, how are they learning these schemas? How are they updating these schemas? So are the schemas represented in the cortex, or are the schemas hippocampally dependent? That's a question that we can ask. And in addition to that, is the new learning and the updating of the schemas, is that hippocampally dependent, or can they update the schemas without a hippocampus? And as we talked about over the last couple of days, the hippocampus is this structure that is known to be important for the encoding of new memories, and some even think that the hippocampus may be important for the maintaining of memories throughout the lifespan. So, to test how important the hippocampus was in this task, the experimenters looked to see, can animals that have had a lesion to the hippocampus, that is to say they've had the hippocampus surgically destroyed, can they do this task? And so here I'm putting up a figure showing that when the animals have a hippocampal lesion that is surgically made 3 hours after new learning. So again, these are animals that have spent the whole 6 weeks learning the schema, learning the map of the place and food associations, and now they've just had this single trial of learning the new A prime and B prime locations. And you can see that if the lesion happens 3 hours after the surgery, then the animals that have had the hippocampal lesion are not doing very well. Their performance is really bad on the new learning. However, if you wait a full 48 hours after the new learning, and then perform the surgery, then the animals with a hippocampal lesion are just fine. And you can see that they completely maintain their ability to navigate the map and to demonstrate knowledge of these place-food associations. And so what that shows us is that the hippocampus is important for the updating of the schema, but the hippocampus is not important for the maintenance of the schema. So the schema must be actually stored somewhere else in the brain, but the hippocampus is important for editing it. And so now the question becomes kind of, how is that process working? How is this editing process working? Does the editing happen? Is the editing completely hippocampally driven? That is to say, when you encounter new information, does the hippocampus kind of unilaterally update the schema and move that new information into the schema? Or does the schema, wherever it's stored, most likely in cortex, do you see the cortex actually driving the change in the schema and interacting with the hippocampus to make that change happen? And so that was the next question that Richard Morris asked in these series of experiments. And what he did for this was to take 3 sets of animals that had only that had simply spent 6 weeks studying the map of place food associations. And he had no new learning for them. They were just animals at the end of their 6 weeks that had performed well, but were not doing any new learning. He additionally had animals that were learning the new A prime and B prime locations. So these are animals that have new learning happening. And then he had completely experimentally naive animals just as a control. And so when you put these animals into the task, what you should see, you would think anyway, and he was looking at prelimbic cortex. So this is cortex that has strong association or strong connection with the hippocampus, and is a good candidate for being the kind of driver potentially of these schema edits, these changes in the schema. And so if you think that the cortical activation is actually driving the editing of the schema, then you should see more activation in this prelimbic cortex you should see more activation in the prelimbic cortex for the animals that are engaging in new learning than for the animals that are just recapitulating the learning that they've already done. And in addition to that, if this is schema editing that's happening, then even though the experimentally naive animals are also doing new learning, they shouldn't show prelimbic activation because they have no schema to edit. So when it's completely new learning from scratch for these animals, then you would expect that they would just have hippocampal activation. And so he looked at immediate early gene activation, which is just kind of a way that experimenters, it's not hugely important, but to understand for the purposes of this, but it's a method that allows experimenters to look at the activation in the neural activation in different areas of cortex as a result of whatever task that we happen to be looking at. And so we're going to take a look at a figure for that right now. And so you can see here prelimbic cortex activation is the dependent measure here, and we have animals that are just navigating the original map, animals that are learning 2 new locations, and animals that for them the map is just completely new. And we see that the prelimbic cortex is showing greater activation for this new learning. And so that's suggesting that the prelimbic cortex is actually driving the encoding of the new information. And we saw earlier in the lesion study that the hippocampus was crucially important for this process as well. So we know that the hippocampus is important, but we know that the prelimbic cortex is getting extra activation for new learning, suggesting that the prelimbic cortex is possibly driving in some way this editing of the schema. So just to kind of wrap up, what this study kind of shows us, or not kind of actually does show us, is that schema learning is a real thing. First of all, these animals take 6 weeks to learn the original map, but then take only a single trial to update the schema, suggesting that they do have this kind of overall representation of the map for place and place food associations. So schemas are real in animals. That's an interesting fact, and it facilitates learning of new information into the schema. We know that the hippocampus is important for the updating of the schema, but the hippocampus is not important for the maintenance of the schema across time. And we know from this final study that the prelimbic cortex actually drives the editing of the schema. So it's not a fully hippocampally dependent action. So we actually have this kind of, you can almost think of it as a top-down driver for memory editing, which is a really interesting way to think about it. And so that's going to be my update for today, just telling you a little bit about schemas and a little bit about this particular study of schemas in animals. And so I hope you found that interesting, and I'll talk to you guys tomorrow. All right, bye.